So welcome back, dear students, for the respiration part 4. So today we are going to learn how most of the aerated plants, roots that are taking place, how the exchange of gases are taking place from by the root hairs in the air spaces as well as in the soil particles.
Ma'am, we are unable to listen the sound of this video.
This part we have already completed, Ramsha. Now I'll be explaining it to you from here. So the aim of the experiment is to prove that carbon dioxide is released during aerobic respiration. Aerobic and anaerobic, you know it very well. Aerobic respiration in the presence of air, and anaerobic respiration that takes place in the absence of air. And we have seen how respiration in yeast takes place. So for that, what we are doing is we are taking 2 plastic glass bottles, germinating seeds, dry seeds, 2 small injection bottles or beakers with lime water. And then what are we doing? We are taking a handful of moong dal. You take either moong dal or the bajra because they germinate very quickly. So we are going to keep them in water for a day to perform experiment. And we keep these soaked seeds in a cloth pouch and tie these seeds. Anybody has done this at home? Anybody has got the experience of doing it at home? Sprouted, when you wanted to eat sprouted moong or chana, this is very healthy, you know. If you just try, every one of you, I want you to try this experiment and show me tomorrow when you see this seeds, germination of the seeds starts, the small baby plant starts growing from the seeds, which you can see. So what you have to do is throughout the night you soak either chana, whatever is available at your home, moong dal or chana, and then green moong, you take green moong, okay? And the next day morning, you keep it into a cotton pouch. You call malmal. Malmal ka kapda hota hai na? You better take that. And you keep that seeds into it and tie that pouch. And after 2 days, you open and see. You can see that the seeds start germinating and they start growing. So now keep the cloth pouch in a corner of your classroom. Now, because you don't have classes, you can observe it at your home. And next day you collect the germinating seeds into this pouch. So what do you observe? You can see this, how these seeds, you know, these tree-like structures that are growing, you can see here. So that this shows that the seeds have started germinating. So now keep it in a glass bottle or a plastic bottle so that you can see it. It should be around 200 ml of the capacity, and take a small injection bottle or a beaker, fill 3/4 of the bottle with lime water. And what we are doing is we are again taking a beaker also, and this we call it injection beaker because we are keeping it, inserting it inside the big plastic or a glass jar. We wanted a transparent jar so that you can observe the changes from outside. And this is small bottle, injection bottle, which we are keeping inside the big beaker. We are filling it with 3/4 of it. You can see it's a measuring jar. So 3/4 of it we have kept as lime water. And can anybody tell me lime water is used to detect which gas?
Carbon dioxide, ma'am.
Yes, very good. If it is the gas released is carbon dioxide, this lime water turns milky. So now what are we doing? We are going to do an experiment to prove that. We are going to tie a thread to the mouth of this small bottle that is inserted inside a big bottle, and we are going to let it hang by the thread close to the plastic bottle tightly, and make similar set with dry seeds. So in one bottle we are keeping dry seeds, and in another bottle you can see germinating seeds. Have you seen that? In one bottle, these are the seeds which we have not soaked and we have not germinated. And now we are going to observe the difference in both the lime water solutions that are kept inside, and keep both the sets undisturbed for 1 or 2 days. During this time, observe the color of the lime water in both the sets. So now you have to observe. So supposing if carbon dioxide is released during the process of respiration, what will happen to the color of the lime water?
It will turn milky white, ma'am.
Yes, it will turn milky white. So now, which one do you observe as turning into milky white? The one which are the dry seeds or the germinating seeds?
Germinating seeds.
Yeah, in germinating seeds, you have observed that the injection jar has changed its color into white milky. So now they observe this beaker placed in the bottle, okay, for 2 days. Then what do you observe? This contains germinating seeds turn into milky white precipitate, and it indicates that germinating seeds liberated carbon dioxide, which turned lime water into white precipitate. Okay, so this proves that during respiration, carbon dioxide is released or given out. So to prove that the heat is liberated, and we are talking about respiration is a process of combustion. We call it a catabolic process. So when this is happening, then the heat has to be generated. So let us check the keep a temperature and check what is happened to the temperature also. Is heat being released during this process of respiration? So to prove that, we are taking again thermos flask, 2 thermometers, rubber corks, dry seeds, and germinating seeds. Both the seeds we are taking. And similarly, again, in the same fashion, we are going to put them into the bell jars and insert them into a one-holed, we are soaking the seeds in a water for a day before the experiment, and keep the same procedure we are going to follow to see that the seeds germinate. Is there any problem you are not able to understand? Next day, we are collecting the sprouts or the germinating seeds from the pouch in a thermo flask, and we are taking dry seeds in another thermos flask. Then what do we observe? What are we going to check now?
The seeds, right?
Now, why are we doing these experiments? To prove
Yes,
that.
to observe the seeds.
Observe what? That heat is released during the process of respiration. So we are inserting thermometers inside it. Okay, one whole rubber cork stopper that closes that. Inside it, we are inserting thermometers. And after that, what are we doing? We are closing the thermos flask with tight fitting rubber corks. So once we have closed it, then what do we see? Initial temperature in both the flasks are recorded. So every 2 hours for at least 24 hours. 2 hours we are going to record. That means 12 times we are going to record the temperature in both the dry seeds as well as in the germinating seeds. And then let us, what should be your observation then? Hence, it is proved that germinating seeds respire and liberate heat, which is responsible for the increase in the temperature. So we observe that the dry seeds, there is no change in the dry seeds because respiration is not taking place in dry seeds. Whereas in germinating seeds, because respiration is taking place and heat is liberated during the process of respiration, you can see that temperature rises. So this proves that heat is released during the process of respiration. Now, what is the difference between photosynthesis and respiration? Let us see. Plants carry out photosynthesis, that they produce their own food from atmospheric carbon dioxide using light energy from the sunlight. This conversion of light energy into chemical energy is used to synthesize sugars from carbon dioxide. Carbon dioxide contains carbon molecules which has got glucose or sugars into it. This process of synthetic anaerobic process which occurs in chloroplast. Inside the chloroplast, anaerobic respiration takes place and carbon dioxide plus water in the presence of sunlight and chlorophyll gives rise to glucose and oxygen. Okay, water molecules and oxygen. So what is happening? This CH2 ON are the minute molecules that gets converted into sugars. But what is happening in respiration? You see that exchange of gases are taking place. The process of breaking down this complex food molecules into smaller molecules, and then this potential energy can be synthesized to liberate heat and energy. So we see that this glucose in the presence of oxygen is breaking down to give you carbon dioxide and energy. So photosynthesis and respiration both appears to be an opposing reaction. They're opposite reactions, but both have very different biochemical pathways and they are essential for the plant's growth. So if you look at photosynthesis takes place inside the chloroplast to produce sugars, starch, and other carbohydrates for the plant's metabolic needs. Whereas respiration takes place in the mitochondria of the cell, and these carbohydrates, they are burned to produce chemical energy to function at the cellular level. So photosynthesis takes place during the daytime because sunlight is essential, and respiration takes place at the night because it does not require sunlight and it's just a reverse process. I said photosynthesis is an anabolic building up process and respiration is catabolic, that is breaking down. So now this ends with your chapter, and if you have got any doubts, please ask. So that we get any doubts, if you have, we can get them clarified. Yes, anything? So now you have completed with your tests. If you have understood, if there are no questions, so let me assume that you have understood it thoroughly well. So start doing your exercises, which are at the back of the textbook, and we'll see if you are facing any problem. So from tomorrow onwards, we are going to start writing all the annexures at the back of your textbook, and if you are finding any difficulty, I'm here to help you. Yes, Ramsha, what was your question? Until then, be safe and take care, good care of yourself. See you all tomorrow in the assembly. I want to see you all in the assembly tomorrow.